All right, let's talk about primary lymphoid organs. So let me write it down, primary lymphoid organs. And in this one, we'll be talking about thymus. And this is the part of the innate immunity course. And I promise you, if you complete this course, you will know everything about innate immunity. So let's talk about primary lymphoid organs. These are the sites where specifically immature, undifferentiated lymphocytes. The lymphocytes that are not matured, that are not differentiated, they are matured and differentiated in these sites, right? So the maturation, and the second point, which is differentiation of these cells, will happen inside these primary lymphoid organs, right? So I'm creating this immune cell right over here, showing you all the receptors are there, and this is our immunocompetent cell. So immunocompetent cell means it can participate in the immune system. And this is happening before the antigen presentation. There is no interaction of the antigen. That is why this phase is known as antigen-independent phase. Everything, maturation will happen before the interaction with antigen. So we have thymus. This is bilobed structure. Second one is, I hope you remember, bone marrow. Bone marrow. These are the two important primary lymphoid organs where you have generation of these immune cells and also maturation of these cells, right? So let's talk about thymus. And this is really important organ. If you see, this organ is present above heart in the thoracic cavity. So let me show you diagrammatically. This is the human body, and there, right over there, is our heart, okay? And above that, we have a small bilobed structure, which is our thymus. And this is, as I told you, it's bilobed in structure. It will have two lobes. If structurally, I want to show you, look at this. So this is the bilobed structure you have, and outside what you have is capsule. And that's the outer covering of this particular organ, right? And now if we go a little bit detailed, detail inside the thymus structure, you will see the structure like this. You will have distinct regions. Usually you have two regions. One is cortex and second one is medulla. You will have outer region, which is the cortex region that you can see with the red color I'm drawing. That is the cortex. So this is the cortex where actually the T cell development happens, okay? T cell development will happen, and the maturation will happen inside that which is inside of the cortex. We have medulla. Right over here, we have mature cells, and very, very few cells will be there. So in this one, you have organ in thymus, you have T cells, you have thymic epithelial cells. And these are structural and functional components of thymus. Then you have macrophages. Role of them is to degrade the dead cells. And then you have dendritic cells. Role is to, play an important role with antigen presentation. You have, next is, let's talk about T cell maturation. T cell maturation, you have those immature T cells that I'm designing over here. And these T cells, immature T cells, they are coming from where? These are the immature precursors. They are coming from bone marrow. So this is the bone marrow right over here. And from bone marrow, these cells, the immature precursors, they are coming right over there. And then where they are going is they are going to bilobed structure, which is thymus, and specifically to the cortex region. So you have multiple different phases. Let's talk about first one, which is cortical phase. In the cortical phase, what you have is rapid proliferation of those cells. You will have lots of those T cells. Cells are going to divide, and you'll have many more of these cells, okay? And they need to be further selected. In these particular cells, you will have TCR, T cell receptor. You will have CD molecules, which are cluster differentiation molecules. And then how they are going to get, formed is using the VDJ recombination. You will have a lot of different types of cells. Next is selection process. They need to be selected, right? So you have to remove the self-reactive T cells. The organ will remove the self-reactive T cells. Almost 90 to 95% of those proliferated cells, they are going to die. And only 5 to 10% of these cells, they are going to pass to the next stage, okay? This is how the selection happens. Now let's talk about a little bit more about the positive and negative selection. So you have different types of selection. Your first one that we'll talk about, let me write down the topic, which is component of the selection. So first one, A right over here is positive selection. Positive selection, that means you have the cells, they are going to get removed, which are, interacting with the self-MSC molecule, or cells, they are going to get selected which can recognize self-MSC molecule. And self-MSC molecule, for example, MSC1 cells, they're going to differentiate into CD8 positive T cells. MSC2, they're going to differentiate into CD4 positive T cell. Next is negative selection. The cells that are self-reactive cells. So let's imagine this is a particular T cell which is reacting with a self-antigen. So self-reactive T cells, they are going to die. And that happens with AIRE, which is autoimmune regulator. And this autoimmune regulator is, going to produce tissue-specific antigen. And because of that, it can actually remove those cells. Next is medullary phase. Let's talk about medullary phase. In this one, you have 5 to 10% cells. They are going to move into the next phase. And this is, let's imagine this is the cell. And this will go to the medulla region, which is the inner region of the thymus. And here, they are going to mature into immunocompetent cells. That means they can generate or they can act in response to the immune system, right? They can act. Now, next is the release phase. In the release phase, what is going to happen is now cells are matured. They're going to come out from thymus and then start working, right? Training is done. So thymus, they're going to get released from thymus. And now it's actually the part where they're going to fight with infectious agents. So mature T cells, they're going to come into the circulation. Okay, so let me design that. This is the blood vessel you're seeing, and now the cells inside blood vessel, you can see they are in circulation. And these are the cells now, they're going to fight the infection, mature T cells. So this right over there, they are in the bloodstream, and then from there, they're going to move to the secondary lymphoid organs. So there is a step-by-step process that they follow, secondary lymphoid organs, right? We're going to cover that also. So now let's talk about significance of this particular process. You have diverse T cells that you're getting after selection, right? You have diverse population of T cells. You have MSC restriction, because then they can recognize MSC molecules. Self-tolerance, they're not going to react against self-proteins, self-antigens. Autoimmune response is also regulated in this case. And you have a lot of naive T cells that are ready to interact with the antigen. All right? So this is the part plays an important role. The cells are coming from B cell, and then thymus is there to actually mature them. And once they're matured, they're ready to fight. All right. So now let's conclude this one and let's move to the next module where we will be understanding the different part of the innate immunity. All right.